Fighting Blindness Canada's View Point Education series shares the latest in vision research, provides opportunities for you to ask your questions, and connect with other people who are living with a blinding eye disease. The session you're about to watch is a recording. To learn more about the research we fund and upcoming programs and events, please visit our website at fightingblindness.ca. Viewpoint is proudly presented by our Platinum partner Bayer and supported by our Gold partner Roche and Silver partners AbbVie, Astellas, Beacon Therapeutics, Johnson & Johnson, and Specsavers. Thank you for watching. We hope you enjoy this session and share your thoughts with us in the comment section below.
We are now going to hear from Dr. Deepak Lamba, but before that, I just want to tell you about a really exciting fundraising event that we're holding in June, and hopefully we can get some people in this room interested in joining us. So we've got an event called Move for Sight, and this is Fighting Blindness Canada's national fundraising event. And it's a way for people across the country to come together to move in their own way. They can walk, they can run, they can do a yoga class, anything to stay active, to raise awareness, and to raise funds for sight-saving research. What makes Move for Sight so special is that it's not just about moving, it's really about community. And so it's really about standing up with the thousands of Canadians who have vision loss and showing that you care about advancing research and supporting those who are impacted. So we really invite everyone in the room to, whether you join solo or have a team, start a team, it's a really great way to sort of take what you've learned here and put it into action. So you can learn more or you can sign up at moveforsight.ca, and we'd very much love to have you join us. And if you have any questions, please feel free to ask me or one of the staff, and we can give you more information as well. And now to get to Dr. Deepak Lamba. So we're so excited to have Dr. Lamba here, all the way from California. That's California, yes. So Dr. Lamba is a distinguished scientist who is affiliated with the departments of immunology, regenerative medicine, and neurosciences at Genentech. He's a clinician scientist with over 20 years research experience working on retinal development, stem cell-based repair mechanisms, and disease modeling with a focus on translational research. And his work aims to bridge the gap between laboratory research and clinical application, ultimately trying to enhance patient outcomes in ocular and ophthalmological health. So thank you very much for joining us, Dr. Lamba.
Just very broadly, I have a bunch of slides that I'll show. I'll try my best to describe them for people who can't see it clearly. But if there is something that's not clear, feel free to reach out to me after the session, and I'll try to explain these things. With that, just briefly disclosure, I'm an employee of Genentech Roche, and so while most of this talk doesn't deal with anything that I've done there since I recently started there, that's just some of my affiliations. With that, I'll come to the main part of what I'll be talking about. And essentially, this talk will mostly focus on the retina, which is the main light sensing part at the back of the eye. And within these eyes, there are a bunch of different neurons and support cells. And for the specific part that I'll be describing today, I'll focus on 3 different cell types. And these are the key cells important for most of the diseases that we talked about today: inherited degenerations, macular degeneration. There's the pigment epithelium, which is at the very back of the retina, and this is the main support cell that keeps all of the retina really functioning well, our light sensing to work, providing nutrition to the light sensing cells, which are the photoreceptor cells. And these photoreceptor cells are the ones which perceive all the light, convert them into a chemical signal that the rest of the body can make sense of. And in addition to that, one other cell type that I'll very briefly talk about is a glia cell. This, for those who can appreciate it, it's this cell in red color. And this is the cell type that comes out from the stem cells. It is not a stem cell in our body, but used to be in other species, and I'll talk about its relevance for some of the things that are happening on the research side with relation to them. But this cell type is also really critical to keep the eye functioning. All right. So with that, what's the retina to what's a stem cell. And as somebody mentioned, like they had never heard about stem cells in this context before. So what are stem cells? And very broadly, what we define a stem cell is based on two properties. One is this ability to self-replicate, to make more of itself. And so that's what defines it being having a stem property, a property that can make something else on top. And then the ability to generate new cell types. This is very context and capacity dependent, and so not all stem cells are the same. Different stem cells have different roles to play. And that's really important to understand just based on what's coming up in the clinic. And so moving more deeply into, so what are stem cells? So I'll divide them into two very broad categories. One is pluripotent stem cells, and so this, by the name pluripotence, ability to make lots of different things. And the classic example is our blastocyst, our inner cell mass from which we as humans are born. So these are cells which have the ability, one, to self-renew, so make a lot of themselves, and then go on to make literally every cell type in the body. And so they can make all the way from neurons to lung cells to kidney cells to pancreatic cells, all of these. So they have the potential to make all of them. On the other side is these adult stem cells. And the adult stem cells are what we have as adults. And these are very restricted in their capacity, and they're very context dependent. And so, for example, hematopoietic stem cells make blood cells. So they can make different types of blood cells. And so when you donate your blood, and your blood levels go down, these stem cells are activated, make more of these in their body. You have an injury, you lose some blood. They will not go on to make new eyes, new brain cells. They are restricted in what they can do. Same thing with skin, for example. You have a scratch, there are stem cells at the bottom, they regrow your skin. And again, the skin cell cannot make something else. Same with adipose tissue. So there are some which can make everything, others which are very restricted in their capacity. In all of this, there is this new excitement in the field on something we call an induced pluripotent stem cell. So this is Nobel Prize winning work from Shinya Yamanaka and Jamie Thompson, who showed that by identifying certain factors, they have been able to, in a dish, convert different cells, like the cells in your blood or your skin, to make them pluripotent. So they can actually be converted to a cell like the cell that was in the embryo that can make other cell types. And this has pretty much revolutionized the field, both in terms of being able to make a lot of stem cells, but also use these stem cells for disease modeling. I won't go into that part today. I'll focus purely on stem cells for therapeutics. But this is an area that is absolutely taking over the field, where we can actually take somebody's cell. So one of the things that I worked on when I was at UCSF was a patient who had inherited degeneration, a severe form of IRD called LCA, due to mutation in CRX. And so we had individuals whose blood we could collect, program them, make them into actually the eyes that they have in a dish, and actually understand what's going wrong, how can we work to fix it. So this field has completely taken off on how we can actually get a better insight of what's happening inside our eyes or brain or any other body part. So I'll just recenter back to stem cells for therapeutics for the rest of this talk. And so, as I said, stem cell therapies will depend on what stem cells they are talking about. So don't assume if somebody says it's a stem cell therapy, that's just going to work for everything. So going back, do we have stem cells in our eye? And as I mentioned, we do not really, as humans, have stem cells. So the key thing that came out that we have spent a lot of years figuring out is, as humans, we've lost our capacity of regeneration, at least in terms of the brain or the eye. Lower species actually can do that. There are things like newts, which I'm showing at the very top of the slide. These critters, you can pretty much scoop their entire retina and they regenerate it completely back, completely functioning retina multiple times over their lifetime. Unfortunately, this capacity has gone down as we've evolved, partly because if we do that as humans, we bias towards getting more cancers because we are much longer lived. But lower species can do it. Things like zebrafish and goldfish have retained some of this capacity from that glial cell that I was showing in that previous slide, this Miller glia, which can, in lower species like fish and in chickens can, to some extent, regenerate either all or subsets of these cells. But as we became mammals, we lost this capacity as for our research that we do on mice, which actually implicates what we see in humans. So as such, is there a way to recover? Can we actually regenerate, or by putting in certain factors? So that's an area. Can we make new stem cells back in the body so the body can fix itself? This is a huge area of research right now. Like I mentioned, since somebody could figure out how to take your blood cell and become almost a pluripotent stem cell, which can make any cell, can we restore that limited capacity of a cell in the eye to restore the cells we've lost? So these are what we call reprogramming factors. Can we program this glial cell back to a stem cell so it can replace the cells that we have? As I said, ongoing intense area of research, but still results are somewhat limited. I'll show one piece of data. This is not work from me, but a mentor of mine, Tom Ray's group up at the University of Washington, where they took these glial cells, which in this figure are these green cells on the left side. These are cells which span the retina. And they put a bunch of these reprogramming genes in there and made them on the rightmost end, like a neuron. And so when they tested its property, it changes the property from a glial cell to a neuron-like cell. Unfortunately, the capacity was very limited. They could never make either a pigment epithelial cell or a photoreceptor cell. So there's still a lot of work that still needs to be done, but at least it shows that there is a potential to do something like that. And so there's a lot of work going on sort of restoring inherent capacity to rejuvenate our stem cells. So moving from what we can do inherently to, if not that, what else can we do? And so what are sort of other types of stem cells that could potentially be used for patients who have retinal degenerations? And here again, I'm splitting them into two broad categories. One broad area of research is sort of adjuvant cell therapies. So here the idea isn't to replace your cells back, but put cells in that can provide support to the dying cells so they can live longer. And so that's what I call on the left adjuvant cell therapies that provide what we call neurotrophic factors, or trophic factors, support factors, which can improve the survival of dying cells. One big thing, none of these are clinically approved. So these are things that are in clinical trials right now. I have two examples up here. One is the CD34 blood cell-derived therapy that's ongoing, a trial for patients who have retinal vascular occlusive disorders. And then another clinical trial by JSite, a company that came out of UC Irvine, where they took fetal stem cells, grew them up in a dish, and are injecting them into the eye to provide support factors. Both of these trials are very early phase, but they have seen some small signals. So here the idea isn't to replace the cells, but at least be able to support the cells which are dying. Again, the idea of as Dr. Benson mentioned, gene-agnostic therapies. Doesn't matter what disease you have, your cells are dying. Can we do something to support these cells to keep them alive longer? On the right side is actual cell replacement therapy. So can we use stem cells to directly replace the cells that we've lost? Here I'll focus on two broad cells, as I said, the pigment epithelium and the photoreceptor cells. Again, no clinical trial. They're in clinical trials, but no clinically approved products. It's very important to mention that all the time. So I'll start with just the cell replacement therapy options, initially with that pigment epithelium, because that's the one area that has advanced the most in clinical research. So here, as I said, just going back to the thing, that's the main cell at the very back of the eye, which supports the light-sensing photoreceptors and is affected in certain forms of inherited degenerations as well as macular degeneration. At least based on my own work, suggests that that's the seat of the disease. That very early on, that's where most of the changes happen. Again, the idea here is for treatment of patients who've lost a bunch of these cells. These aren't patients who can receive therapies very earlier on, but at least towards the late stage where you've lost these cells, you may need to replace them back. Hopefully you can find factors that can rescue earlier so you never have to do a stem cell therapy. Even though I spent my entire life researching it, my hope is that someday we actually never have to do any of these, because putting new cells back in is very, very difficult. This is not easy therapy. But there always will be patients who have late stage of disease where you may have to do some of these. So going to stem cell therapies, the pigment epithelium, as you said, it's pigmented cell, and people have now figured out ways to make these in large batches. So what you're seeing in the picture on the right is a flask which is to the brim made up of these pigment epithelial cells. This flask, for example, will have about 120 million cells. Most cell therapy trials that are ongoing require only about 150 to 200,000 cells. So a single flask like this can treat hundreds of patients, and these can be grown in the lab. So at least the research has reached a point where people have been able to make these cells in a dish in very high efficiency. The next step is how do we deliver, and those are, again, huge challenges, but there are clinical trials ongoing. Here's a list of some of them. There's, like Dr. Benson mentioned, there are lots of trials ongoing and keep coming in. These are some of the key ones which are happening either in Northern America or up in Western Europe, but there are trials elsewhere as well. The people are using these in different ways. So there are two ways that people are trying to deliver it. Either deliver these cells as a suspension, so you can just go in and inject these cells at the back of the eye where they're lost, or others who are trying to put them on small patches, so you can put a small patch at the back of the eye. And so I'll go through pros and cons of each of them a little bit in the next slide or two. So one of the trials is a trial by Lineage Therapeutics that Roche is partnering on. And here the idea is, can we be making these cells in a suspension that you can inject into the back of the eye? And that's the cartoon shown behind me. So you have cells where in patients with end stage of AMD called geographic atrophy. So this is end stage where you've lost a large chunk of your pigment epithelial cells, and because of that, your light sensing photoreceptors aren't functioning optimally. And so can we replace the pigment epithelial cells, they can start supporting photoreceptor cells. That's the idea behind this clinical trial. Again, it's an early stage clinical trial. I think right now it's in phase 2A, but they're seeing small efficacy signals even in these trials. So this suggests that there is an opportunity for these cells to potentially help patients. Again, very early stage trial. It's not something that's in the clinic, but it suggests that there is a chance that we can make these cells and they may have some efficacy. There are still a lot of challenges to figuring out how best to deliver, how to make sure they stay in the right spot. Will these cells have an immune reaction? So I'll go through some of those challenges towards the end that a lot of folks are still trying to work on. But the field is really moving forward to a point that there are things that are starting to go into patients. So that was a way where people are trying to put it as a suspension. Here's putting it as a patch. And the reason for trying to do it as a patch is because you're trying to put it in an environment where disease has caused the layer where the cells need to sit to undergo atrophy. And so some people think that may not be enough to, even if you have good cells, sort of, if you put good seeds on a bad soil, they'll never take. And so are there ways that you can create an improved soil in that sense? So that's the idea behind the patch. Can you have a patch where you already have these cells on top, and if you have these cells on top, then you can just slide it in the area that you need. The challenge is sliding a piece like this in the back of the eye. You're putting it in a bad environment. So there are a couple of trials that are ongoing, one using this on the left, a patch which is about 6 millimeters by 3.5 millimeters, because that's still really big for the eye to make a cut around so you can put a layer back in. But the trial's ongoing, and I'll show some data from that. Because of the challenges with excuse me, that trial, National Eye Institute ran a different trial where they made a much smaller piece. So this is only about 2 to 4 millimeters, so helping a very small patch of your eye, but still a little bit more doable in terms of trying to slide this at the back. And so these trials are ongoing. There's not much data yet out on the National Eye Institute trial, but I'll show a little data that has happened on the regenerative patch technologies trial. So here's the data from that trial. What you're seeing on the left or my left, your right, I guess. Yeah, no, your left too. Is sort of the area of GA that's highlighted, so area in the central retina which is dying over time, and here they went in and put that patch in. So I said it's like 6 millimeters, a really tiny patch, but it looks huge in this picture because we are just focusing on that central part of the eye. And these cells, at least in the trial, could at least survive for up to 4 years without causing any severe reaction. So there's a potential these cells can stick around long enough. But when they followed up these patients, the results have been somewhat mixed. So what you're seeing on the table is how many patients where they saw some improvement versus stabilization versus worsening. The last one's what you don't want things to worsen. But if you can even stabilize the disease, it's still an improvement over what's happening. So the trial's ongoing. They're still trying to figure out how you can improve on some of these cells. So this is one of the longer running trials that have happened on the patch technologies. All right, now I'll briefly move on to photoreceptor therapy trials. Let me just make sure I'm staying on time. Okay, yeah. So this is now talking about the light sensing cells. So this is the area that I've spent 20-something years trying to figure out stem cell therapies for light sensing cells, because essentially we are losing our vision because we're losing our light sensing cells. So can we figure out a way to put those back as well? And so, as I said, we've spent a fair bit of time, and that's not just my research, but research across the world. A lot of folks have now figured out to make these light sensing cells. So on this side, you're seeing these balls of cells, and that ball is actually a mini retina. So if you took out a piece of retina from a human eye or a mouse eye, it would look exactly the same. But this is made out of stem cells that we reprogrammed from blood from a patient. So that just shows how much the field has improved. We can actually make what looks exactly like what's in the eye. In B there's what I've highlighted in red is the region of the light sensing cell where that light sensing property happens. These outer segments, that fuzzy area is where light's converted into a chemical energy. So they actually look exactly like what's in the eye. We've done some work where we tried to put these cells back in patients who had, in mice who have a patient mutation. These mice are born completely blind from birth, and in these mice, we could show small evidence of rescue of vision. In C is looking at an ERG, an electrical signal in the eye, and where we could see small improvements. And when you're looking at the whole eyeball images, in that, what you're seeing is the ability for the pupil to constrict because it can actually recognize lights coming in. So it looks like these cells have the ability to go in and survive and make some connections. We don't know what that vision means. Very difficult to figure out in other species, but at least it shows preliminarily that this could work in patients. Again, as I said, this is just some of my work. There's a whole field that has worked on it, including Dr. David Gam, who recently got a trial just approved, and I'll briefly mention that. These things have started to go in patients. So this is data from a clinical trial that's happening in Japan by another researcher called Masayo Takahashi, who has started this work around the same time I did, so about 20 years of work on her lab side. And in Japan, they've been able to run a very limited trial. So this was a trial in just 2 patients, where they could at least take that mini ball of an eye, the retina, they shaved out the part that looks like photoreceptors and transplanted that in patients. And what you're seeing is at least these cells survive for about 104 weeks, so a couple of years, good survival, staying in the area that they should be. Unfortunately, they didn't see a huge improvement. In one of the 2 patients, they saw some stabilization of vision with minimal evidence that might be something happening. The other patient did not change it much. So very early phase, a lot of work needs to be done to figure out how to get these cells to work, how to make them connect, a lot of these things that we need to work on. And then, as I mentioned, Dr. David Gam, who was funded by the Foundation Fighting Blindness and And the Canadian counterpart of the foundation, their work finally just got FDA clearance to do a phase 1 clinical trial for photoreceptor therapies. Again, lots of work, lots of years spent on trying to make these cells optimize what you want to put it in, and this is some work that would lead to trials in the United States. So what are the sort of key unanswered questions around it? So what are the key challenges around cell therapies? And I sort of mentioned a few as I was giving this talk. What's the best way to deliver them? We still don't know. As I said, some people are trying these suspensions or dissociated cells. Others are putting in patches. Others are trying these mini organoids. We don't know what would work best in a patient. That's what the clinical trials are for, to really get into what could potentially work in a patient. We can get as much as we can think of we can get from lower species, but the human trial is the human trial. That will really give us better confidence. Just how much mature these cells need to be. Do we need to be fully formed retinas, or do we need to be just enough so they have the plasticity to actually still integrate in a bad environment? As I said, the environment is challenging. That's where some people are trying to put patches because there is inflammation, kind of like when you scratch your skin, it's red. In our eyes, as degeneration happens, it's constantly red. And so you have to start thinking about how do you get new cells to survive in an environment which is not healthy for them. So there's a lot of work going on trying to figure out how to improve a microenvironment which would better take these cells. The disease state, as I said, right now, most of the trials are for end stage because, again, these trials are run on patients where you cannot do more harm and you don't want to do any more harm. But eventually we need to figure out what's the right timing when we could best help the patient. And then, again, will they make the right connections? These are things that we'll know once we do it in a patient. I want to end by something that I was talking to somebody else earlier today, and to me is very important to discuss. These are these rogue clinics, because everybody keeps hearing about these stem cell clinics in Europe, in the US, all around the world, essentially. Here's a map of all of these unregulated clinics, and this is an old map, actually. So there are all these clinics that say that they've figured out how to treat patients, be it for eye disease, osteoarthritis, all sorts of things. And the box on the panel on the right, maybe my right, is what sorts of cells are being injected in patients. Anything from fat cells to bone marrow-derived cells to even amniotic fluid-derived cells. So people are putting a lot of things without any real evidence for it. And to me, it's not just malpractice. It can actually have bad effects on the people that eventually it will not even allow them to have better therapies. There was a very critical sort of point that happened around 2017 where there was a clinic in Florida where they said they have a clinically tested product that could work, and they injected fat cells in these patients, and all 3 of these patients not only lost their vision completely, but some of them had to have their eyes enucleated. So this is just like, be very, very careful. There's a lot of this out there, a lot of misinformation out there that you need to be very careful about. And it's not you. It's like this was a survey done where asking people what they think. If you find something online, what it means. And a lot of people just assume if somebody puts it on their website that it's on a clinical trials site, it is approved, and it is absolutely not. You have to be very careful what you do. Foundation is a really good place to ask about all of these things if you find something online. Here's just some gotchas that are always there. A listing on clinicaltrials.org does not mean that it's actually a clinical trial that's approved. The trial design, literally it's just a listing site. You could put something on there that you're running a trial as a company and you can just list it. It's just a listing site. It's not an approval site. And a lot of these trials just say they are FDA compliant, which again means nothing. It's not an FDA approved. You have to make sure you're reading through the text, understanding what these words mean. If anytime you're unsure, talk to people in the foundation, talk to your doctor. They will give you the real advice. Just be very, very careful about this. At least for me, that's the most important takeaway. If nothing else went through from my talk, just be very careful. So I'll just end by summarizing what I said. So essentially, I think stem cells have a huge potential to rescue vision. While local regeneration is currently not possible, there's a lot of research going on to figure out rejuvenating our stem cells in a way. This, to me, there was a mentor of mine told me the best stem cell therapy would be one where you would never have to inject anything to the eye, because you would not have any of the challenges that we are facing right now of trying to figure out how to make them work. These non-retinal stem cells have the potential to support, but none of them are either clinically proven, and they carry a lot of risks, so be very careful. And finally, pluripotent stem cells work is actively going on cell therapies. These have the best opportunity to completely restore vision, but they need to be clinically validated. And with that, I'll end and happy to take questions.
Getting blinded. Thank you so much, Dr. Lamba. I think just something to add on about that sort of PSA about rogue clinics, especially if it's saying it's a clinical trial. You should never have to pay for a clinical trial, whether it's stem cell or any other treatment. If it's a legitimate clinical trial, the company will pay for you to travel, they'll pay for the treatment, so you should never have to pay out of pocket. So that's another way to sort of be cautious about it. Okay, I can already see there is a question in the audience right up here. Morgan is coming behind you to give you a microphone.
Would you mind putting up your first slide? What is a stem cell?
Sure.
Yep, the question was about what is a stem cell.
Sure.
Okay, well that was easy. Other questions in the room? Okay, we've got one right over here, and then we'll go to the back.
Behind you here.
Yes, Dr. Lamba. Blue Rock Therapeutics and Nanogen, I believe, is running a stem cell clinical trial. And they were looking for late-stage retinitis pigmentosa candidates, which I am. But when I went to the website, they said the age limit was 12 to 50. Can you explain that, why that is?
So, I'm not an expert on clinical trial design, but usually the clinicians that are part of these trials figure out what they think would be an ideal age where they could actually see some signal. So often a clinical trial design would be somewhat limited on that, wouldn't be what actually goes into a patient eventually. They often broaden it, but for a trial design, because you don't want the trial to fail, they figured out where they think they could have the best improvement, so that way they could move across clinical trials. That would be my understanding, but somebody from that company would probably be able to clarify better.
Okay, thank you.
Absolutely.
My question is, if you're blind from birth, are you hoping the stem cells will fix that?
So it should not matter when you started losing vision. The only wrinkle to that answer is how much your eye has changed over time. So once you start losing your cells, what happens is the rest of the retina starts reconfiguring itself to try to make some sense of what's happening. And so as you progress, sometimes it reaches a point where they've made too many misconnections within itself, and that's where new cells may not be able to completely fix it. But that's an assumption right now till somebody comes in. The flip side to some of this is our brains are absolutely amazing. So sometimes the brain can start making sense of small signals. And the example for that being There were a couple of clinical trials which are not stem cell based on these bioengineered chips that they were putting in the back of the eye. This was these Argus devices that got approved in Europe, and all they had were like between 8 and 64 electrodes, so that many points of stimulation, and the brain could still make some sense, like edges, contrast differences. So there is some evidence that the brain may still make some sense out of it, but you will never rescue full vision in that case. Yes, absolutely.
What is the state of retinal transplant?
So initially there was some retinal transplants in terms of fetal tissue, that they were taking whole pieces of retinas and transplanting it. That part of research has almost stopped now, so there aren't any clinical trials using fetal tissue anymore. But these mini organoid-based stuff or dissociated cells are sort of what's taking over that step of transplant therapies. Yeah.
The idea with the stem cell replacing the photoreceptor is confusing to me because there are there are cones and rods, and within the cones, aren't there 3 types of opsins?
Yes.
So you could end up with a stem cell replacing, if it did work, you could end up with just one opsin that would make it all red. Everything you saw was red or green or blue.
Yeah.
Has the theory of vision changed? So all these photoreceptors can be duplicated as if they're the same?
So two parts to that answer. So one, what we make in a dish, we actually make all the different cell types. So we make the rods and we make all types of cones. The flip side, even if you're making or only a subtype of these cones are surviving, it's not that you would be biased to a certain vision. So essentially how we make sense of colors is by comparing which part of that circuit is in it. So if that's what happens in patients who have this color blindness, all they lose is one type, and what they see is sort of this grayish vision around them. And so you may not see perfect vision, but I think currently the idea is with stem cell therapies, can we rescue some useful vision for these patients? Patients who not able to see, not do normal tasks. If you can rescue some amount of that they can be somewhat independent, I think that's a huge win. So that's where the initial premise is. Eventually we would want to do all of what we are saying, perfect vision, but that I think would take much longer. Right now the idea is just get enough vision back to some of these patients so they can at least have some normal vision functions around the house, not bump into walls, things like that.
My question is, how will the recent introduction explosion of artificial intelligence affect the research community, and how will it affect your research from a timing perspective, and how will it speed it up?
So there's a huge, as you point out, huge opportunity with AI to test out either hypotheses on how things are changing by accumulating large pieces of data together. For example, at Roche, we are spending a lot of effort on AI to create what we call foundational models, a model that could start predicting things that are going wrong. A little away from stem cell therapies, but what else can we identify using this that could rescue patients or restore some amount of function back by modulating different pathways. So there's a whole lot of work that's ongoing to find new targets that you can try to fix things. Some of that predictive analysis could be applied here, where you say, okay, what's the critical barrier to getting these to patients? And so there's a lot of work going on for sure. Yeah.
Okay, I think time for one small question, if there's a short one. And if not, Dr. Lamba, I think, will be around for a few minutes, so you can ask him some questions, come up and chat with him afterwards. And I am going to have one more thank you for Dr. Lamba. Thank you so much for joining us.